Applying artificial intelligence to help discover new medicines could become one of the more transformative ways we use this tech in the near term. And one of the key players in this space is a company called Recursion. This week, I'm excited to get back to my reporting roots and take us on a field trip to their labs. If you haven't joined us for one of these visits before, think of it like the grown-up version of Mr. Rogers visiting a crayon factory. It's a chance to peek behind the curtain at some of the cool work that's happening in science and tech. I'm Jennifer Strong, and this is Shift.
Now here on the steering column is a device called Auto Cruise. You simply set the speed you want.
Self-driving robo-taxis are already on the road in 2 U.S. cities.
As the disc rotates, a mirror reflects the light in the way that depends on how the signal was recorded.
This is the 100 terabyte optical. I present to you Electro, the Moto Man. Ladies and gentlemen.
I would say that one of my greatest skills is my ability to interact with humans.
This episode, we head to Salt Lake City, Utah, where we tour the labs of Recursion Pharmaceuticals.
From Zuckerman Spader. For 5 decades, Zuckerman Spader has been a leading litigation firm trusted by in-house and outside counsel, businesses, and individuals. With offices in Washington, Baltimore, New York, and Tampa, Zuckerman Spader serves clients throughout the country and around the world in high-stakes disputes, enforcement matters, investigations, and at trial. When the lawyer you choose matters most. Online at Zuckerman.com.
My household's love for Quince started with a silk dress. It's lightweight, comfortable, and always on point. The surprising part is everything with Quince is priced 50 to 80% less than what you'd expect to find for premium fabrics and finishes at similar brands. By working directly with artisans and cutting out the middlemen, Quince gives you luxury pieces without the markup. And for me, the best part is that Quince only works with factories that use safe, ethical, and responsible manufacturing. We own a number of their items, including school polos for my kids and summer weight cashmere. The craftsmanship and quality have consistently been off the charts. Elevate your closet with Quince. Go to quince.com/shift for free shipping on your order and 365-day returns. That's QUINCE.com/shift to get free shipping and 365-day returns. Quince.com/shift.
This is a multi-trillion-dollar industry. Many of these are hundreds of years old. Like companies like Merck and others have been around for a very, very long time. And about a decade ago, there was a set of 6 to 10 upstarts that wanted to use things like ML or AI to try and help discover medicines better. Today, there's probably 400 or 500 of these companies, most of them still very early stage. And we, Recursion, are one of the very few of that original wave of companies founded in the 2010s. We were founded in 2013. So I'm Chris Gibson, co-founder and CEO of Recursion. So Recursion is a company that is trying to make sense of this massively complex thing we call biology. So inside of each of us, we have trillions of cells. And inside every one of those cells, we've got about 20,000 genes that are expressing about 400,000 different proteins. And there are trillions of interactions of all of those 400,000 proteins. So it is like wildly, wildly complex. And that's the problem. We don't understand it well enough to make medicines at a rate that could get us essentially inexpensive medicines. So today, 90% of drugs that go into clinical trials fail. And it's not because there aren't really smart scientists who are working on the problem. It's because they're working against this incredible complexity. And our belief at the founding of Recursion was that if we were going to take our pipette, which is this little thing you use to move liquids back and forth to do experiments, and we were going to do sort of manual science, it was going to take us thousands of years to generate enough data to really understand how it all works. And so instead, we envisioned a company where we could use robots to generate massive quantities of data, and we could use machine learning and AI to take all of that data and try and distill it down to find the patterns that actually tell us how biology works, so that we could go from failing 90% of the time in clinical trials to maybe failing 80% of the time in clinical trials. And I tell the team, if we could go to 80% failure rate, we'd be twice as efficient as the industry. And you could theoretically cut the price of medicines in half if you were able to do that over and over again. And that's going to 80% failure rate. Imagine a day where it's 50% failure rate. And then one day in the future where we can completely model biology in silico, where you can identify and predict the exact medicine for each disease, or even one day each patient, synthesize it, validate it, and go right to the right patient. And all of a sudden, one day in the future, we might have a 90% success rate. And that's ultimately the multi-decade journey that we and others are on, is to try and decode biology so that we can eventually radically improve lives. One of the funny stories about where we are right now, we're in an old Dick's Sporting Goods, and we converted that into a biotech lab. That's why there's a giant rock wall behind you. I was wondering about that. We did not put that in. That was here when we got the space. We also learned a lot about retail slabs, and they're only 6 inches thick, and they vibrate too much. So we have $400,000 worth of steel in the parking garage underneath our lab to keep it from vibrating, because that's actually a big issue for doing these experiments. We're walking up to our main Phenomics lab. And so before we go inside, I'll tell you just a bit about Phenomics. So people have heard of things like gene sequencing, and Phenomics is actually very similar, but instead of looking at what kind of gene or RNA or protein is in a cell, we're actually taking microscopy images of cells and then using computer vision to turn those into mathematical representations of biology. So like, take a picture, it's worth 1,000 words, and for us, it's worth a whole bunch of data about what's happening in a cell, like what disease state does it have, or what is a drug doing? And we can generate up to 2.2 million experiments worth of those images every week in the lab that I'm about to take you into. So take it all in. All right. And here we go. Yes.
Okay. Entering a gown room.
Yes.
Okay. So we're going to
right,
suit
so
up.
We're going to go ahead and gown. All right. Let's head into the laboratory. So you are entering the largest Phenomics lab in the world, that I'm aware of at least. What I find fascinating is that this facility, full of robots, a handful of people, it's doing the equivalent of my entire PhD's worth of data generation about every 15 minutes, which is simultaneously exciting and depressing, given that I sat there doing that work for those 5 years. We're going to walk through, but just take a minute to look a little bit at this, because you see like all of these different workstations, all of these different lights, little arms moving plates around. And this system is running essentially like 20 hours a day, 7 days a week, 50 weeks a year. It's really more like an advanced manufacturing facility than it is like a traditional research lab. But that's one of the shifts we're seeing in the world right now. We're moving from sort of doing data generation with a pipette using your hand sitting in a laboratory in a kind of low throughput way to generating extremely high quality, extremely high scale data in a highly, highly automated way. And so why don't we walk through?
And just to kind of set the scene here, there are half dozen people in here at once? Is that typical, the 50 weeks a year you have people too, or?
Yeah, I would say if you take us out, then yeah, roughly 6 people. I think that's about right. And it goes up or down a little bit. There are times when the lab can run completely autonomously, but also it's not completely error-proof, and the experiments are quite expensive. And so we'll have people around ready to jump in if something goes wrong. So one of the things that I'll point out that's kind of interesting, you'll see that we have like ring or nest cameras above each of these work cells. So we actually record every movement of every robot. So if later on, or even in process, our QC system detects that we've run into an issue, we can actually go back and understand what happened. Like, did a plate fall down? Did somebody put something in the wrong way? And we've actually created like this set of instant replays for the team during training, where they can see all the mistakes that a person might make and learn why our procedures are able to help us avoid making those mistakes with video replay. It's just like a sports team. You can see on some of the screens here, we have almost what looks like Gantt charts and a variety of different dashboards. We are tracking all of the different metadata from all of these different pieces of equipment so that we can ask and answer questions like, how long was every reagent on every individual experiment? Because when you're running millions of experiments, you can't do everything at the same time for every single experiment. You have to kind of do them in order. But biology is really sensitive. And so if you add like a CRISPR-Cas9 reagent and it's 20 hours on one cell and 21 hours on another cell and 36 hours on another cell, if you're not tracking those sorts of things, you'll get wildly varying data that you won't be able to make sense of. And one of the real innovations of what we've been building for the last decade is a laboratory where we measure everything. So when we go build machine learning models, we're not actually building a model just on the biology, but actually taking all of this metadata in as well so that we can make, essentially get higher signal and understand some of the noise. And this is one of the problems with the public data sets that are out there. They're great. We're glad they're out there. But very often, this kind of metadata that we collect, every single movement of every robot for every single well of every experiment, we don't always get that data. In fact, you almost never get that data.
Actually, can you take a pause here and just explain that for someone who's outside of pharma, has no idea what you're talking about in terms of public data and how this gets used and what you have to do to make it useful?
Yeah, so there's lots of data that goes into understanding biology. Scientists are doing millions of experiments all around the world, and the vast majority of that data sits within given academic laboratories or companies. But sometimes people or governments will put big chunks of this data out for other people to use. So, for example, in the United Kingdom, they've sequenced hundreds of thousands of people and have put that data out in a safe way to allow various researchers who have permission to come in and get access to the sequencing data in the hopes that that'll help them generate really, really exciting discoveries. Another great example, people have maybe heard of AlphaFold. It's this protein folding tool that's very, very cool. That was built using a public data set that had been generated over the last 40 or 50 years. And a set of scientists like 40 or 50 years ago got together and said, why don't we just collect all the data in the same way, record it in the same way, and put it in one place? And the team at DeepMind and now Isomorphic leveraged that data to build this AI model that helps us understand how proteins are folding. That's a great example of a success with public data. In general, though, we think that there's probably like 0.1% of all of the biology and chemistry data out there in the world that's public. And as I said before, sometimes it's missing a lot of those key measurements that help you make sense of it and really understand what's signal and what's noise. And so that's why we decided at the beginning of Recursion that we were going to have to build a laboratory to build the data the way we wanted it. And today, the data we've built here and the data we've partnered to bring in, we're north of 60 petabytes of data, which is like many libraries of Congress worth of data. It's a massive quantity of data.
Do you ever browse the web in incognito mode? If so, it's probably not as incognito as you think. If you read the fine print, you'll see that all your online activity is still visible to a ton of third parties, unless you're using a VPN. ExpressVPN hides your IP address, making it extremely difficult for third parties to track your online activity. It reroutes 100% of your traffic through secure, encrypted servers, so third parties can't see your browsing history. Plus, it works on all devices, phones, laptops, tablets, and more, so you can stay private on the go. My family's privacy is really important to me, and we've tried a number of VPN products, including this one. ExpressVPN is easy to use. You just fire up the app and click one button to get protected. Protect your online privacy today by visiting expressvpn.com/shift. That's expressvpn.com/shift, and you can get an extra 4 months free. expressvpn.com/shift.
So over on your left, you'll see our test bed of our next generation of platforms. You see a bigger set of robots underneath this rail system that has these little, almost like little magnetic train cars. And if you wait just a minute, you'll see one. These are carrying plates around. And so rather than having a set of robots in one place that does one specific thing, like add CRISPR-Cas9 or add a drug or take pictures of cells, all the robots to do all of the things we want to do are in one place, and we can move plates and experiments to any of those robots in any order. And so we're testing this out before we build out a bigger version of that in the future. And on your right, you see the largest collection of high throughput microscopes that we're aware of in the world. So each one of these microscopes is several $100,000, and it is able to take really, really high resolution pictures of cells using different fluorophores, using bright field. We can keep the cells alive in some cases. We can do confocal imaging. And those cables that you see going up to the ceiling, those are full of all of the imaging data and metadata that we're collecting that's running to the cloud and to our own supercomputer, which we keep at a secret location somewhere in the valley. There you go. There's one of the little magnetic plate holders that are going along. Actually, we'll go over really quick across the street, and it'll mean degowning and regowning. There's so many cool things happening in these labs, but they actually don't look that awesome. A lot of the people are doing the cool stuff in the back. We grow massive quantities of cells like neural iPSC cells, which are really, really hard to grow. So we take things like skin cells and we reprogram them to be things like neurons. We had to do this as part of our collaboration with Roche and Genentech. And in the process of delivering that collaboration, we became what we think is the world's largest producer accidentally. It was just what was necessary to achieve. So the laboratory we're going to go into in just a minute is called our compound management laboratory, and it's where we have millions of potential drugs, so little chemicals that are sitting in this giant refrigerator that we can take out to run against all of these different experimentals. So take them and measure them in cells using these images or using gene sequencing or transcriptomics, and we can get a sense of what they might be doing, and maybe one of them could become the starting point for a new medicine. Gowning again. All right, this laboratory is going to have a lot of sounds, so be ready. Some of them are loud. Okay. Although you have headphones on, so that should be okay. So what you see here are a whole bunch of robots. Some of them look like the ones you saw in the last laboratory that are entirely set up to take any one of these millions of chemicals we have inside of what looks like a wall. It's actually the side of a giant refrigerator with a giant robot inside of it. And if we go around to the other side, you might be able to see that robot working. We'll see. All right, we're going to look through this window and you'll see what looks like it almost looks like a scene from The Matrix or something. There's all of these rows and rows and columns of shelves that have little plates full of little tubes, and those little tubes each have a different chemical in them. And there's this big gantry system in the middle that you can see now moving towards us. You'll hear it in a minute. That's actually going through and picking any one of those tubes and customizing plates with hundreds or thousands of these chemicals that we can take back and test on cells. And we'll be looking for one that might make cells that have a certain disease look healthy or act healthy again. And this is kind of the premise of what we're doing at Recursion, building these giant maps of biology and then training giant machine learning or AI models on top of those to try and understand what's really happening underneath it all, to decode biology. And this is the interface where the scientists can collect all of these different tubes. So we see a plate here and you can see, we're putting that one in. Yeah, just to show you a little bit what these tubes look like. Here's one. So we have these little tubes. Inside is a chemical, and then there's a little tiny 2D barcode on the bottom. It looks like a mini QR code about the size, half the size of a pencil eraser. And that's how we know what's in every single one of these tubes. And I've put it back in the right spot. But even if I didn't, that little code would help the system know and correct the error. And then we'll walk over here. I told you before about the laboratory where we generate millions of microscopy images of cells. This is another laboratory where we generate millions of examples of RNA sequencing of cells. So we'll take cells and look at the instructions that those cells make from their DNA that encode what proteins they're going to express. And so we have these Illumina sequencers over here, and we'll take samples, some of them that we actually used in our other lab. We got images off the microscope, and then we take the same cells and we look at the RNA sequencing that comes off as well. So this one's a little bit less exciting looking. These Illumina sequencers are very, very self-contained, but they're generating huge quantities of data as well. And then we're taking all of that data and adding it to the imaging data and other data we generate to try and understand how it's all working. And our hope is that all of this work turns into insights that are going to lead to medicines. And right now we have multiple programs that are in clinical trials, and we can't go on a tour of all of that. That's happening in different sites, different hospitals all around the world, in the US, in Europe, where we're testing some of the medicines that were discovered in these sorts of laboratories in people to ask, are they actually having the effect we hope they would? And are they having any other effects that we hope they don't, some of the side effects? And so we're constantly doing those clinical trials as well. And we have over 100 people that are involved in making all of that happen, but not part of the tour today.
This episode was produced by me and Emma Cillekens and mixed by Garret Lang, with original music from him and Jacob Gorski. Thanks for listening. I'm Jennifer Strong.
RX.